Good day, this is the Professor, CBMD PhD, and today's Monday, August 29, 2022. It's 4:42 and 55 seconds, a.m. Eastern Standard Time. And today's research, cancer. So let's hop to it here. The therapeutic viruses help turbocharge the immune system against cancer. Combination of two therapies shows promise for treatment-resistant cancers. Date August 27, 2022. Source: Arizona State University. The researchers propose a new line of attack that shows promise for treatment-resistant cancers. The immune system has evolved to safeguard the body from a wildly diverse range of potential threats. Among these bacterial diseases, including plague, cholera, diphtheria, and Lyme disease, and viral contagions such as influenza, Ebola virus, and SARS-CoV-2. Despite the impressive power of the immune system's complex defense network, one type of threat is especially challenging to combat, and this arises when the body's own native cells turn rogue, leading to the phenomenon of cancer. Although the immune system often engages to try to rid the body of malignant cells, its efforts are frequently thwarted as the disease progresses unchecked. An illustration shows a cancer cell center surrounded by immune T cells augmented with an oncolytic cancer-fighting virus. A new study describes how a combination of immunotherapy and viral therapy using myxoma virus provides new hope for patients with treatment-resistant cancers. The study describes how oncolytic viral therapy, a technique using cancer-fighting viruses, can act in concert with existing immunotherapy techniques, boosting the immune capacity to effectively target and destroy cancer cells. Oncolytic viruses represent an existing new avenue of cancer therapy. Such viruses have the remarkable ability to hunt and terminate cancer cells while leaving healthy cells unharmed, as well as enhancing the immune system's ability to recognize and terminate cancer cells. One such virus, known as myxoma, is the focus of the current research, an area of expertise for the research group. The study shows that the use of T cells infected with myxoma virus can induce a form of cancer cell death not previously observed. Known as zoonoses, this form of cell destruction may be particularly useful against solid tumors that have proven treatment-resistant to various forms of cancer therapy, including immunotherapy alone. And this work affirms the enormous potential of combining viral therapy with cell therapy to treat currently intractable cancers. The immune system is composed of a range of specialized cells designed to patrol the body and respond to threats. The system is involved in a ceaseless arms race against pathogens, which evolve sophisticated techniques to attempt to outwit immune defenses, propagate in the body, and cause disease. The cancer presents a unique challenge to the immune system, as tumor cells often lack the identifying cell features that allow the immune system to attack them by distinguishing self from non-self. The cancer cells can further short-circuit immune efforts to hunt and destroy them through a range of evasive strategies. Researchers hope to help the immune system to overcome cancer's notorious tactics of disguise, developing new experimental techniques belonging to a category known as adoptive cell therapy. And such methods often involve removing a collection of cancer-fighting white blood cells, known as T cells, modifying their seed and destroying capacities, and re-injecting them in patients. The two forms of adoptive cell therapy, immunotherapy, are described in a new study, CAR T-cell therapy, and T-cell receptor engineering. The basic idea, in each case, is the same: treating cancer with active T lymphocytes extracted from the patient. The development of these therapies has been nothing short of revolutionary, and some cancer patients facing grim prospects have made remarkable recoveries following the use of immunotherapy. But techniques like CAR T-cell therapy and T-cell receptor engineering nevertheless have their limitations and are often ineffective against advanced solid tumors. In such cases, cancer cells often manage to evade destruction by T-cells by downregulating or losing the surface antigens or MHC proteins that T-cells use to identify them. The new study highlights the ability of immunotherapy when it is coupled with viral therapy to break through the wall of cancer resistance, specifically using myxoma-equipped T-cells. The myxoma can target and kill cancer cells directly, but more usefully can induce an unusual form of T-cell directed cell death known as hotasis. And this form of cell death augments two other forms of programmed cancer cell death induced by T-cells known as apoptosis and pyroptosis. During myxoma-mediated hotasis, cancerous cells in the vicinity of those targeted by the therapy are also destroyed in a process known as bystander killing. And this effect can considerably enhance the dual therapy's aggressive eradication of cancer cells, even in notoriously hard-to-treat solid tumors. A combined myxoma immunotherapy approach therefore holds the potential to turn so-called cold tumors, which fly under the immune system's radar, into hot tumors that immune cells can identify and destroy, allowing CAR T-cells or T-cell receptor cells to enter the tumor environment, proliferate and activate. The researchers are at the edge of discovering newer aspects of the myxoma virus and oncolytic viral therapy. In addition, these findings open the door for testing cancer-killing viruses with other cell-based cancer immunotherapies that can be used in cancer patients. The ability to radically re-engineer oncolytic viruses like myxoma to target a range of resistant cancers provides a new frontier for the treatment of this devastating disease. And this is the professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.